Hello everyone, I'm Dr. Chandrasekhar. I'm the Medical Director of Displays Autism Research Institute. Today I'm going to present you the importance of gut health in autism and gut microbiome transplant for autism and how to prevent autism in your second child. So we all know the incidence is rising since 1960, 70. So it was 1 in 10,000 and now it is 1 in 36 in US and 1 in 54 in India, and it's increasing every year. So all these days, we have been thinking that there is no established cause for autism. We kept saying it is multifactorial, but recently we have got a Maha report from the US government, that is Make America Healthy Again. This report says the bioaccumulation of the environmental chemicals are manufactured chemicals and man-made chemicals as the culprit for the autism. So example like pesticides, weedicides, preservatives, food colors, industrial toxins, heavy metals, etc. All these we are taking it from our food, water, air, and they are entering into our body and they are getting stored. The storage of all these chemicals is called bioaccumulation. So this is the main culprit and the primary reason for autism. So how these chemicals are causing autism is this also mentioned in body burden study. The US government has given this assignment of establishing causes of autism to Environmental Working Group in 2005. They studied the umbilical cordlet and they identified several chemical compounds, some 287 chemical compounds in their in that umbilical cordlet. And out of that 287, 180 are the cancer causing, like carcinogenic, and 208 has the ability to cause birth defects, and 217 are toxic to the brain. And these chemicals that are bioaccumulated, they cross the placenta and enter into the babies when they are in the womb. And they even these chemicals has the ability to cross the placenta, and also they can cross the immature blood-brain barrier of the fetus. The fetus is not the fetal brain doesn't have a proper blood-brain barrier. It is immature by that time. So all these chemicals are entering into the brain through the immature blood-brain barrier. So and they have a poor excretory mechanisms, and all this is happening in the very critical windows of the neurodevelopment. So you can see all these chemicals in this picture are entering into the babies. This is the reason. This is the primary reason for autism. And these chemicals, whatever entering into the babies, they even disrupt the important microbiome that which is supposed to be vertically transmitted from the mother. That means which from the mother to the baby through amniotic fluid is also impaired. So this is we have done a careful review of the autism research, through which we understood this is the pathways that children are getting autism, and so these chemicals are and genetic susceptibility are the primary causes for the autism. What they do is like these chemicals, when they enter into the babies, they enter into the neuron cells and brain cells and everywhere, and they start interfering with their gene activity. That means their gene activity. So whatever, so the brain for development and functioning, the gene activity is important. That gene activity is interfered with these chemicals. This activity, this is called epigenetic errors. So because of this, the brain microarchitecture while developing becomes impaired. Like microarchitecture means like the brain looks like normal only. Like it doesn't look, it cannot be picked up in the CT scans or MRIs, but in postmortem brain tissue studies, you will be able to identify the abnormal structure, like the microscopic structures are abnormal, and it is confined to certain areas like higher functions. And this chemical bioaccumulation of the chemicals also disrupt the mother's gut microbiome. And also when they enter into the fetus, the first 1,000 days of the first 1,000 days of the baby is important for the microbiome development. This first 1,000 days starts from the day of fertilization, zygote formation. That means when the fertilization starts during the intrauterine life, and after that 2 years is important for this microbiome development. During that time, a lot of chemicals are accumulated in the baby. That is the reason they will not get the important essential microbiome from the mother womb. This is the causal factor for their gut issues and immune function, immune dysregulation, and many other functions are impaired because of this abnormal microbiome. This microbiome also has ability to alter the gene function. Like microbiome has influence on our genes. The microbiome means like we have lots of microorganisms or intestine. So they produce certain chemicals, certain metabolites that enter into our blood, and they can circulate in every part of the human body, and they enter into the every cell, and they can switch on or switch off or regulate gene activity in all the cells. So when this microbiome is abnormal, so our entire body epigenetic pathways also go abnormal. Because of that, in the brain, a lot of functions are not a lot of functions are suboptimal, and we can call it as dysfunctional. So in this with the epigenetic errors, whatever the microarchitecture abnormality that was developed inside womb, that is incurable and irreversible. So this part cannot be cured, and this part stays for forever. And the brain function, because of the abnormal microbiome, is so the dysfunction, whatever the dysfunction developed due to abnormal gut microbiome, can be reversed by replacing the microbiome. So when the children receive this abnormal microbiome, it triggers sort of inflammation in their gut, and with the age it keeps on progressing. And when they reach age of 15, 16, it becomes more stubborn, and it will be very difficult to treat at that time. So, so these are some of the epigenetic errors generally we see in autism. So I'll just name them. I'll not go deep into this one because we have done it in the previous webinar. So there is a differential gene expression patterns when compared to the normal individuals, and there are differential alternative splicing patterns and attenuated cortical pattern. These are the some signature epigenetic errors that are seen in the autistic brain during developmental phases. There are certain processes that are going abnormal because of this abnormal gene function. So they are neuronal migration, differentiation, and accelerated neurogenesis, microglial dysregulation. So all these actually they cause the microarchitectural abnormalities, and they ultimately lead to a defective wiring kind of thing, like defective functional circuitry. Because of that defective wiring kind of thing, they have multiple behavior abnormalities and everything. And because of this defective functional circuitry formation, so they even land up in virtual autism or aggravation of symptoms when they keep on watching mobile. So these are some of the brain areas that the colored areas you can see. All these areas have this abnormal microarchitecture. So you can see these are the areas you can take a screenshot or something like that. So all these are very sensitive, and human brain is these higher functions are very sophisticated, and so they are very sensitive during critical window periods. When the chemicals enters into these brain cells of these particular sophisticated and sensitive areas, their microarchitecture development is going abnormal and resulting in communication deficits and social impairment and repetitive behaviors. So you can see all these structures this is thickened ventricular zone and abnormal the brain tissue developing in the abnormal areas. So these are important microarchitecture abnormalities that we usually see. Today we mainly focus on microbiome abnormality. So bioaccumulation of the chemicals in the mother's body, first they disrupt the mother's gut microbiome. Mother's gut microbiome is also important for the baby's development because we all most of the, nowadays most of the gynecologists also talking about endocrine disrupting chemicals. They can disrupt the hormonal hormones. They can disrupt many other functions. When these chemicals are accumulated in our body, when they are stored in our body, uh, they can disrupt our microbiome. And when microbiome is disrupted, multiple functions are affected. And one of them is baby's neurodevelopment as well. So these mother's gut microbiome metabolites are important for the fetal development as well. When mother's gut microbiome is affected, the fetal development is also affected. And when these chemicals, bioaccumulated chemicals, enter into the baby, and in these first 1,000 days, they will not allow important microbiome to colonize in their body, in their gut. So this is the major reason we see most autistic children have gut symptoms and allergies and everything. So these children receive a weak, susceptible, and a poor microbiome. So this microbiome abnormality is responsible for all the epigenetic pathways, abnormal epigenetic pathways, and brain functions as well. So when these children receive weak and susceptible microbiome, so when they get exposed to the antibiotics and vaccines, and this microbiome disrupts further, and the microbiome is directly linked to the brain chemical balance as well, like neurotransmitter balance. When this microbiome disrupts, brain chemical balance also disrupts. Then the children land up in regression. This is a regression. We always see that when we give antibiotics and vaccines at the month of 18th or less than 2 years, they land up in regression. This is a quite common pattern we generally observe. So these are the biofilms that we usually observe in the stool of autistic children. So these are so every human being will have a mucus kind of thing on the gut lining. So but they have a different ecosystem in their gut because of the chemicals. They did not get a important microbiome. These children will receive a bad microbiome. So because of this bad microbiome, their mucus forms thick strands kind of thing. So you can see all these thickened things. So these are all biofilms. They have a bad gut bacteria. So this is so clearing out this microbiome is very much important because the chemicals are neuroactive metabolites produced by this bad microbiome will enter into the circulation, and they create a lot of havoc for these children. They become aggressive and they start biting, they start hitting, and other behaviors also, self-injurious behaviors are and many things are linked to this bad microbiome. So clearing out this bad microbiome and repopulating with a new microbiome is the best solution according to our research. So so for this, people are exploring diet and probiotics and microbiome transfer or fecal microbiota transplant across the world. So you might have seen this study that was published in the 2019. This was done by Arizona State University. They have run microbiota transfer therapy for autism, and they observed 80% improvement in the gastrointestinal symptoms and 20 to 25% improvement in the behavioral symptoms. And they also did some animal studies. So they have taken human gut microbiota that from the autism spectrum disorder, and they have transplanted to the mice, and they observe. That means you have seen this kind of biofilms, right? So in the microbes from this, they have transplanted to the pair of germ-free mice. Pair of germ-free mice means this pair means one is male and one is female, and they have transplanted the gut microbes to germ-free mice. Germ-free mice do not have any gut flora. These They have transplanted the autistic children's poop into these germ-free mice, and they observed their offspring. So once when they observe their offspring, uh, the mice harboring this ASD microbiome uh, exhibited ASD-like behaviors, and they also studied their gene activity, their metabolites activity. They have seen it is different from the normal. The mice received the normal microbiota from the normal children. So in that, what they have done is like in this particular study, they have taken two groups. One group of one pair of mice received microbiome from the autistic children. Other group of mice received from the normal children, and they have observed. So they have seen there are they have observed ASD-like behaviors in this mice in the offspring of the mice that received microbiome transplant from the ASD child, and which was not observed in the other group. And they have seen the abnormal epigenetic patterns and metabolite patterns in in this group one. And when they identified there are two important metabolites like 5-AV and taurine, and they have given those metabolites to these mice that received ASD microbiome, and they saw improvement in their behaviors as well. So this was one of the evidence that their microbiome itself is a causal factor for the autism. So this study, you can take the screenshot for this study. This is an important study that will give entire picture of autism. So replacing this gut microbiome. So how to replace this microbiome? So these children are not receiving essential microbiome from the mother because of the chemicals. So how to replace this one? So we have two sources here. So the essential gut bacteria that has to be replenished in the colon, that can be come from amniotic fluid or healthy child poop. These are the two. These are the only two sources right now to replenish. So we getting amniotic fluid is difficult, and using amniotic fluid is also difficult. Uh, so uh, so the poop transplant or fecal microbiota transplant or gut microbiome transplant may be the best solution for this. So this particular procedure is called gut microbiome transplant. So in this, what we generally do, we identify the healthy children and we collect their poop and we test them and we collect their poop and make it a solution and transfer that solution into the affected children's large intestine. So the way you are seeing here in this picture, it is similar to that. So uh, there are other methods also. People use healthy children poop or healthy donors poop, and they take it as a capsules and they swallow it. So, but this is less effective because stomach acids and pancreatic enzymes will kill 99% of the bacteria in that, and only 1% to 2% reach the target organ. But we have a direct access through the anus to the target organ. So that can be done through colonoscopy and retention enemas. Colonoscopy has one advantage that we can visualize the colon and we can see the proper bowel cleanse pattern. And if the bowel cleanse is there, then we can infuse the solution into it. But it cannot be done on a daily basis because the microbiome transplant doesn't work like that. If you give it for once or twice, it won't work. So it needs a long-term, like a very long course and high proper bowel cleanse every day and a long course, and it should be done in a regular approach kind of thing, like 2 to 3 months we have to do. So colonoscopy is not advisable for longer periods, like only one session or maybe colonoscopy may be better. So we came up with retention enemas. We tried with retention enemas. We use a longer tube and we infuse that solution deeper into the colon, and we do a colon irrigation before doing the procedure. And once the entire stool is washed out, we can transplant this solution and paint. So the aim is to paint the entire surface of the colon with the healthy children poop. So a good donor and a proper bowel cleanse of the recipient followed by FMT, and for a longer course with high frequency, like every day, it's like consecutive days, is the key for success of this treatment. So the important thing that we should focus in this particular treatment process is the donor screening. So the process of GMT is important for the success of the treatment. So there is something called the International Stool Banking Consensus. So they have given a battery of tests. So a very big battery of tests are given by International Stool Banking Consensus. So with that, we have to identify the donor. But here the entire world believes in high diversity and they take adults as donors. But I believe in essential microbiome that comes from the mother womb, and whatever the microbiome that is colonized after birth, I consider it is only commensal and it is not an essential microbiome. I have a logic for this because if you take a hypothetically healthy newborn, he is completely healthy, and all his biochemistry and biochemical pathways are normal state. If at all any gut bacteria that has a role in their health, they must be present in the healthy newborn. So this was the idea we led to essential microbiome. So we started looking into the essential microbiome. Also, we have found some important insights into it, and we identified some important strains in that microbiome which are present in amniotic fluid and meconium, which can be which can be considered as essential microbiome. So we take children as donors because children will have that essential microbiome, and we have a lesser risk because adults have the duration of exposure to the chemicals and antibiotics will be more with the growing age, and also there is high risk of sexually transmitted diseases, and sexually transmitted diseases have long incubation periods. So all these things are a bit risky. So I believe children are the best donors, and we use children as donors less than 12 years, and we also do a food intolerance test for them along with whatever the test suggested by the International Stool Banking Consensus. And I'll explain about why we are doing food intolerance tests for the donor in the next coming slide. The process will be like every day stools will be collected in a hygiene environment and in a gamma sterilized container, and it is transported to the clinic at 4 degrees centigrade. And once the microscopy is done, we do a homogenization of the specimen and do a refrigerated centrifuge and use that solutions as a colonic infusions. Like we do retention in mass kind of thing with that solution. So this continues for several weeks. If we do it on a proper ball cleanse on daily basis, and this new microbiome will colonize and they start working. So this is the food intolerance test. This is an important test that it's an important revelation for us in our clinical research. So it's like it acted like this particular test. We understood that this is a litmus test to identify the responders before treatment only. So in our observation, every child with food intolerance responded. Every child who didn't have food intolerance never responded to the treatment. And this also worked for us when in identifying the potential donors also. So those who had there is no food intolerance for them, they became the better donors, and the children receiving also responded well to those donors. So it is the food intolerance is like so I'll tell you why this food intolerance test is needed. Food intolerance test means this is actually so our body is showing some inflammatory response to the foods that we are taking in. So if we have a bad microbiome or poor microbiome, it will read our food as antigens, and it reports to the immune cells that reside beneath the gut lining. So the cellular structure you are seeing here is the gut lining. So over the gut lining there is a bacteria here. So if there is a good bacteria, it will not report our food as antigen to the immune cells. So if you have a bad microbiome, it will report our food as antigens to the immune cells. Then if you have a poor microbiome, you will have food allergies and food intolerance. If you have healthy microbiome, there is no food allergy and food intolerance. This is a quality indicator kind of thing. And another test we can do to test whether the microbiome is good or not is stool microbiome test. People do it for with the 16S RNA sequencing or a shotgun metagenomics. Currently, it is a little inconclusive. So we use food intolerance test as the quality indicator. And in future, we are developing this control database of the amniotic fluid and meconium database. So then this particular test will become conclusive after that. So this is the process we usually do in our clinic, like uh, the collection from the collection point of collection to the administration. It is done very diligently, and the stool collection from the children will not happen in a restrooms or anything. It will be done in a hygiene room in their donor's house. There will be a potty seat, and these like we give sterile grapes to them and sterile cups, which were sterilized using gamma radiation. The child will pass the stool in it, and once the child is pass stool in it, and it is the stool is submerged in the saline, and air tight lid is placed, and it is kept in the refrigerator in their home, and the our unit people collect it in 4 degrees centigrade and bring it. In the processing unit, we use the homogenizer, and we don't use blenders kind of thing, and we use a homogenizer so that there is no least air entry and there is no harm to the anaerobic bacteria. And we use a refrigerated centrifuge as well for the supernatant fluid. So this is the method we use, and we prepare two different types of solutions. One is viscous solution, one is supernatant solution with the refrigerated centrifuge. These two solutions are used for colonic infusions. So what are the benefits that we can expect? So the first thing that we can expect is the gut symptoms. So most children will have constipation, diarrhea, and poor stool quality, improper digestion, and they have colitis, anal fissures, and etc. So we can over time through to once we start doing FMT for a few months, then over 2 to 3 months, all these gut symptoms resolve, and the food allergies, food intolerance, and they also get reduced over time, and we can see it in the food intolerance test or food allergy test. Serum Ig levels and CRP, ESR, all these things will reduce after FMT. And so children with autism, they have this immune dysregulation. They have yeast infections and SIBO, all these things because of these are all opportunistic infections. After FMT, there is an immune modulation, post FMT immune modulation is there. Because of that, these opportunistic infections get controlled, and their digestion absorption improves. With that, their functional deficiencies get addressed, and their hormones and enzymes also get into the homeostasis with FMT. And the brain chemical balance. Children with autism will have this neurotransmitter imbalance. So once we do FMT through the microbes, once they colonize there and survive there, they start producing the neuroactive metabolites that enter into their bloodstream and enter the brain, and they switch their neuronal pathways and in the neurons and everywhere. So then there is a change of neurotransmitter balance through which their irritability reduces and their sleep and mood disturbances get better, and cognitive development. There will be some cognitive development, and their sensory issues and fine motors improve, and their learning capability improves. And these are the important things we have seen with FMT. So like we talked about, the chemicals are the primary reason for the autism, and these chemicals disrupting the mother's gut bacteria is the secondary cause kind of thing. So we understand how we can prevent autism also through these pathways. Whatever the research we have done, we understand autism is preventable. So when if somebody has a child with autism, when they are going for second child, they must be careful because there is a 18% chance of autism in the sibling. So we must screen for the inheritance pattern, like mother, father, and baby. We have to do a genetic testing and do the screening, and then we have to identify. We have to detox the mother before pregnancy and maintain the detox status throughout pregnancy. And mother's gut health must be priority, and they should maintain that particular they should maintain the gut health throughout pregnancy through diet and probiotics. Through if we do this particular all precautions, we can prevent autism in their second child. Those who are looking for second child after first child is autistic, they can go through how autism is occurring and what are the causes, and so how we can remove all these bioaccumulated chemicals. So figure out how to remove chemicals and reduce the exposure to the chemicals throughout pregnancy, and have a safe have a safe pregnancy and avoid autism in the next child. So these are some of the chemicals like so you can see. So we use this nail polish and sunscreen lotions, cosmetics. Fish also has this heavy metals kind of thing. So the non-stick cookware. This is the most important. So perfluoride alkyl substances that are present in the non-stick cookware is the biggest problem, and pallets and parabens and plastic, pesticides and herbicides. These are the chemicals that uh, that can enter the baby and can create a problem during neurodevelopment. So we should avoid exposure to all these chemicals during pregnancy. Thank you. You can ask questions. I'll answer for all the questions.
This is Vinita. I have a question. So, once this whole procedure is complete, what are the chances that the gut microbiome will go back to its original level or again um, be affected and it's not as good
Yeah.
anymore?
Yeah, so it is possible if somebody doesn't maintain a diet and bowel cleanse during treatment protocol and after treatment also. If somebody is taking sugars and junk food, processed food, all these things, and bowel cleanse is also not proper, then they can go back and their microbiome, the existing microbiome is very strong, so it can bounce back and they can come to baseline if the diet and bowel cleanse are not maintained. So maintaining diet and bowel cleanse is important during treatment. After treatment, also 3 to 4 months we have to maintain. So it's very difficult to get rid of their existing microbiome and repopulate the new microbiome. That's why they take 2 to 3 months of time.
Okay, and the people who are like outside India, how much time do you think they should consider staying or the process? Is it like 2 months, 3 months?
It's it's
Um,
it's 2 to 3 months is the best time. At least 2 months is the best time. Like because for to see the efficacy of the treatment, 2 weeks or 3 weeks is enough. But to get the sustained result, 2 to 3 months is necessary.
okay.
Doctor, this is Sobia. You talked about how these toxins are causing epigenetic, that is, they are switching the genes
Yes,
to create symptoms of autism.
yes,
So, once you do the microbiome transfer, is the possibility there to switch that gene back?
yes.
And, okay.
So when the brain structure is getting formed, genes also switch on and switch off, and they produce proteins, right? When the structure is formed, the whatever the structure is formed during that time, the genetic epigenetic errors happen. So then there is a microarchitecture abnormality that cannot be reversed. But the brain during brain function, the neurotransmitter production or signaling pathways, they their signaling pathways and epigenetic errors in those things will be reversible. So all the signaling pathways, neurotransmitter balance comes after FMT. So we can see some cognitive improvement and learning improvement and improvement in the sensory issues and fine motors. We could see improvements here, but complete recovery is not possible because the microarchitecture abnormality that happened inside the womb is irreversible almost. So very little improvement will be there in that microarchitecture abnormality.
Okay, so you're saying with the microbiome, if there is brain architecture issue, but
Yes,
even with the microbiome, there you're going to see some improvement at least
yes,
with
yes,
signaling pathways. Though
yes,
you might not get like 100% improvement or reversibility, but you're going to see a difference in the
yes.
child. Okay.
Yeah, microbiome has a role in the epigenetics in entire body. So once the structure is formed, it cannot repair and like it cannot reform the entire structure, but it will improve the brain function through epigenetic pathways only.
Hi, doctor. This is Shruti here.
Uh,
I would like to know, is there any age barrier for a child to take this, or if the child is also having other medications along, is it possible to do this treatment for him?
yes, medications will not be a problem. So age is like first few years is the best time to do it. Like first 6 to 7 years is the best time to do it, but it can be done at any age. With the age, they have, like I said earlier, when they have a bad microbiome, there is some sort of non-specific colitis in their body, in their colon, which is subclinical, slowly worsen over time, and they become irreparable over time. So, if we are doing it in the older children also, we do see some improvements, but the improvements will be in the gut tissues and the hormones and other things, but there won't be much benefit in the brain aspect in the older children. So, in the first 10 years, as the first maximum is like we have seen best improvements less than 7. 7 to 10 also we have seen improvements in the brain function, but with older children we didn't see much improvements. Uh, uh, Considering if you are talking about brain function, older children did not get much improvement with FMT, uh, but they also got other improvements like overall their allergies reduced and their gut health improved, their hormones improved. Uh, they became comfortable and their medical issues resolved.
Okay, thank you. Just one more question. What I mean, my child is 8.5 years old right now, and
Okay.
he's having cognitive issues. He's having autism with ADHD.
Okay.
So and when we look at it for 2 to 3 months, he's going to school. So we need to. I mean, I'm not in Hyderabad or the place that you're doing this, right? So is it possible to extend a little bit during his holidays to do it, things like that? And what's the cost factor involved around it?
Yeah, it's cost is not a big issue because it's being done in economical way in our institute, and we can extend your time, so it's not a problem.
Okay, great. Thanks a lot.
So we have one question, sir. Uh, uh, the question is, could you please explain a bit more on how GMT helps in achieving hormones and enzyme homeostasis?
So hormones and enzymes also they are proteins. So their production depends on their gene expression patterns. Children with autism have a different ecosystem. They have effect on their enzyme production, their pancreatic exocrine function, and many other endocrine functions also. Their production is the production of these hormones and enzymes is affected due to differential gene expression, that is epigenetic errors. So when this microbiome is replaced, there they get a balance of those enzymes and hormones. It is through epigenetic pathways.
Okay, we have one more question. Good morning, doctor. If my child is not positive for food intolerance test, FMT will not work for him, right?
Yes. If
Okay.
there is no food intolerance positive on the blood test, they will not respond. Because it is like food intolerance positive means they have a bad microbiome. Food intolerance negative means it's a good microbiome. So microbiome replacement can be considered when there is a bad microbiome. It is as simple as like somebody is deficient in iron, we'll replace with iron injections or something like that. Somebody is deficient in vitamin D, we will give vitamin D. Somebody doesn't have a good microbiome, we give good microbiome. That's it.
Okay, we have one more question. What type of cleansing is necessary for mothers to do in order to eliminate these toxins?
So we can do some sort of glutathione injections and vitamin C injections. There are some supplements like N-acetylcysteine, astaxanthin. So we have to take this all this antioxidant therapy 3 to 4 months prior to pregnancy. Then remove the chemicals. Then throughout pregnancy also vitamin C, vitamin A, vitamin E, and all other antioxidants can be taken so that there is no chemicals entering into the baby.
Okay. Any other questions? If anyone has, they can ask directly by unmuting themselves or maybe write it in the chat section.
Dr.
Hi,
I do have a question. So my kid is 5 years old. We have already done multiple rounds of stem cells and we have seen the improvements with his allergies, with his sleep behaviors. Everything is sorted out. Right now, my concern is more around the comprehension and functional understanding of things. And then, expressive language is what I'm looking at. So how much this FMT is going to benefit him?
Yeah.
Knowing the fact that whatever you spoke about in the your neurotransmitters or whatever you have improved, it is all about sensory and
Yes.
behaviors is what you talked about.
Yes.
But
So
can we expect further improvements in his these areas?
Yes, you can expect improvement in the cognitive ability. Cognition is a diffuse function of the brain. Once you achieve the neurotransmitter balance, their cognitive ability improves, their learning capability improves, their comprehension also improves. So, if the language areas are properly developed and speech areas are properly to develop to some extent, if some children already had some improvement in already their brain areas of the speech and language areas are developed to a certain extent, they will improve a lot in the area of language and speech also.
How long have you been doing this, the FMT, and what's the success rate?
So we have been doing it since last 4 years. Yeah, we have been doing it for last 4 years, and the success, the revelation is like this: like if somebody has food intolerance positive and gut symptoms and regressive autism, they improved, and it is like somebody has food intolerance, all of them improved. Those who didn't have food intolerance positive, they never improved.
Okay, we have one more question. 1 second. Hi, when my kid was at the age of 1.5 to 3 years, he frequently affected with fever, and to recover, sometimes it takes 1 week. That time, doctors prescribe antibiotics, then only he recovers from fever. Does it causes autism? Now he's okay with the health, no fever issues. His current age is 5.7 years.
Okay. So if autism starts in the first trimester of the pregnancy. Somebody can lose microbiome after that. If the brain structure is developed inside the womb properly, they will not land up in autism. If somebody's microbiome is brain development got hampered in intrauterine stage, they only develop autism. So all antibiotics and vaccines aggravate the problem. Became autistic by birth inside the womb itself, and they receive a susceptible and poor and weak microbiome that can be disrupted with antibiotics and vaccines. When it happens, their existing neurotransmitter balance also goes for a toss, and they lose the skills. So this is regression pattern. So these antibiotics and vaccines or some any other environmental insults can they can aggravate the problem of the autism. Not they do not cause autism directly.
Okay. Yeah, please ask the question. I'll
Yeah.
proceed with.
Doctor, I have I have two questions. I'll quickly go at them. My son, he's 7 years, 4 months now, or 6 months. He has mold and aspergillus mold in his gut. And so my understanding is this should resolve that as well.
Yes.
And the second question is, in the West, Tylenol is given very frequently. Like it's almost second, like giving candy to the kids when they are sick. Does Tylenol or acetaminophen have any impact
So,
on autism?
yeah. So you must be knowing that microbiome has it has role in drug metabolism as well. So if you have a bad microbiome, Tylenol cannot be properly metabolized. Then they can create a problem for the child. And not only Tylenol, many drugs can create a problem for them when they have a bad microbiome. And the second, and the first part of your question was Aspergillus infection in the gut. So any fungal infection or any SIBO or anything, they are opportunistic infections when they have an immune dysregulation. When you do antifungal therapy and FMT, then so the immune system gets modulated over time, and our immune system starts fighting against that fungal infection, and they can recover completely from the fungal infection.
Doctor, hi. I also have one question. I wanted to know when we did some tests, there was some high toxic level in my son's body. He is almost 12. So, doing this FMT, will the toxicity also reduce and help him to recover fast?
So detox pathways also link to the epigenetic pathways. So microbiome will open up those detox pathways, but because of this bioaccumulation since over a long time, so we have to actively remove those chemicals by doing detox treatments. Like you can take glutathione injections and other things, vitamin C and other antioxidant therapies. You can remove all those chemicals and replace the microbiome. That would be the best thought process for the child.
Okay, doctor. Thank
Can,
you.
can I ask top of that question, Doctor?
Sir, hello? Sir, hello?
Hello?
Just let's do it one by one. So I think Mr. Kabin can talk right
Yeah.
now, and then the next
Yeah.
person, please.
So the top of the previous question, I have one more question. But when you have a detox heavy metal issue, so is it you're saying, sir, like we have to clear the heavy metal before come to FMT? That's one question. And the
Okay.
second question you have that you say that mention older kids, it's very tough to get the treatment going and get some results. So my son is of course going to be 11 and a half right now, 12 going to be very soon. So we did a bunch of stem cell and etc. So is it, if I bring him, is it I will get some positive results out from you? Like especially my son is only have an issue with the communication and understanding the concept, right? So it's basically there's no any other food allergy or anything else.
Okay.
So it's that those two issues I have it right now.
Yeah. So the thing is, so you just need to do a blood test for food intolerance test. If they don't have a gut issues and if their overall health is good and there is no food allergies, you just check for their food intolerance. If they don't have food intolerance, this treatment will not help the child. If they have a food intolerance positive on the blood test, you can expect some changes at the age of 11 or 12 also.
Okay. But about the detox, we have to clear before come to FMT?
So it can be done parallelly. So there is no mandatory kind of thing that it has to be done prior or something like that. So it can be done parallelly. So you can, but detoxing can be done. You can start it before coming also, because simple antioxidants you can start taking astaxanthin or vitamin C, vitamin A, glutathione. All these things you can start at home before coming to the treatment.
Okay, sir. Thank you. Sure.
Yeah, there's the follow-up question. Is it helps to reduce EOE?
What is EOE?
Hello? Sir,
Yeah.
hello?
Honestly, I don't know. Yes, sir. Yes.
Hello?
Somebody, yeah.
Yeah, please.
సార్ ఇప్పుడు ఈ ఫుడ్ ఇంటర్నెట్ టెస్ట్ ఒకసారి అది రెడ్ ఎల్లో అది ఏమన్నా ఎక్స్ప్లెయిన్ చేస్తారా? అసలు రెడ్ వచ్చింది మార్క్స్ ఏంటి, ఎల్లో ఏంటి, గ్రీన్ ఏంటి?
So it is అంటే ఇప్పుడు what you are seeing red, yellow and green patterns in the food intolerance, right? So red ones are highest. So there is it is the extent of immune response. Like if the foods that are red in the come into the red zone, you have to reduce in their food intake. So you can take yellow and green zone.
అంటే ఏంటి అడగట్లేదు అన్నది సరిగ్గాని.
సో అది ఇమ్యూన్ సిస్టమ్ అనేది ఇట్ ఈస్ ఫైటింగ్ అగైన్స్ దట్ ఫుడ్ అని దాని మీనింగ్.
ఆహా.
మన ఇమ్యూన్ సిస్టమ్ ఇట్స్ ఫైటింగ్ అగైన్స్ దట్ ఫుడ్. కాబట్టి మనం ఆ ఫుడ్స్ తీసుకున్నప్పుడు ఇన్ఫ్లమేటరీ ఫ్లేయర్స్ ఉంటాయి బాడీలో.
ఆహా.
రెడ్ జోన్ లో వచ్చిన ఫుడ్స్ తీసుకుంటే బాడీలో
ఆ.
ఇమ్యూన్ ఫ్లేర్ అప్ ఉంటుంది. ఐజీ లెవెల్స్, ఐఎస్ఆర్, సిఆర్పి లెవెల్స్ పెరుగుతాయి అనమాట.
కాదు సార్, ఇప్పుడు మన ట్రీట్మెంట్ ఇప్పుడు ఇది టాబ్లెట్స్ రూపంలో ఏమి ఉండదా?
టాబ్లెట్స్ రూపంలో ఏముండదు అది. కొన్ని ప్రోబయోటిక్స్ ఉన్నాయి, అవి తీసుకోవచ్చు.
ప్రోబయోటిక్స్ వాడాలా అంటే పౌడర్ లాంటివి అండి?
ప్రోబయోటిక్స్ అని ఉంటాయి కొన్ని టాబ్లెట్స్ లాగా, అవి అవి వాడొచ్చు కొంతవరకు, కానీ ఫుల్ ఫ్లెడ్జ్డ్గా దాంతో ఎక్కువ రెస్పాన్స్ రాదు.
ఓకే సార్. కాదు సార్, ఇప్పుడు నాకు ఒక చిన్న డౌట్. ఇప్పుడు మనం డ్యూరింగ్ ట్రీట్మెంట్ ఫుడ్ డైట్ ఏమైనా ఉంటుందా?
డైట్ అంటే మెయిన్లీ మనము అందరి పిల్లలకి రిక్వైర్మెంట్ ఉండదు. కొంతమంది పిల్లలు బాగా ఇష్యూస్ ఉంటాయి, కొంతమంది బాగా సివియర్ గట్ ఇష్యూస్ ఉంటాయి. వాళ్ళకి ఏంటంటే ఫుడ్ ఇంటాలరెన్స్ టెస్ట్లో ఉన్న రెడ్ జోన్లో ఉన్న ఫుడ్స్ని తగ్గించాలి. అండ్ దానిలో ఇంకొంతమంది చాలా పిక్ ఈటింగ్ ఉంటుంది, వాటిలో లిస్టప్ చేసుకోవాలి ఏ ఫుడ్స్ తింటారు అని. అండ్ వాళ్ళకి డైజెషన్ ఇష్యూస్, బ్లోటింగ్ ఇష్యూస్ ఎక్కువ ఉన్న పిల్లలకి లో ఫాట్ మ్యాప్ డైట్ యూస్ చేయాల్సి వస్తుంది. సో అది దాని ఒక చైల్డ్ టు చైల్డ్, ఒకవేళ ఇన్ కేస్ కొంతమంది చిల్డ్రన్కి డైట్ కూడా రిక్వైర్మెంట్ ఉంటుంది, ఇట్లా స్పెషల్ డైట్. అండ్ మోస్ట్ ఆఫ్ ది చిల్డ్రన్కి అందరికి మాత్రం షుగర్ కానీ, ప్రాసెస్డ్ ఫుడ్ కానీ, జంక్ ఫుడ్ కానీ ఇవన్నీ తీసుకోకూడదు.
సార్ ఇప్పుడు ఒకవేళ ఇన్ కేస్ అదే మీకు మొన్న మలహోన్ ఇచ్చాం కదా సార్, ఒకవేళ మేము ట్రీట్మెంట్కి వస్తే కనుక ఇప్పుడు ఏదన్నా అక్కడ పీజీ హాస్టల్స్ అయి ఉంటాయి కదా, వాటిలో ఆ ఫుడ్ తీసుకొచ్చి అంటారా?
మీరు సొంతంగా చేసుకుంటే బెటర్.
సొంతంగా చేసుకుంటే బెటర్ అంటారా? ఓకే సార్. ఓకే సార్, థ్యాంక్ యూ.
Sir, one more
So,
question.
yeah.
The colon treatment that you mentioned, the way to do it in a colon treatment, is that going to be a couple of times in the 2 to 3 months frequency, or is it just one time?
It's every day. It's every day for 2 to 3 months. That means it will be done on a daily basis for almost 2 to 3 months. Around 90 to 100 times we will do it in that 2 to 3 months.
Okay. And you mentioned that is the best way, and it's rather than having the capsules, right?
Yes, yes. Because capsules have very little amount. What we do in general, like when so we have done multiple types of strategies and we understood, we figured out. So every day, what we do, we infuse the saline and irrigate the colon and do enema. So the once the stool is completely evacuated, then we infuse the solution deep inside, like 100 centimeters inside, with a small thin tube. It's painless. But even if it is coming immediately out through from the intestine, it will paint the entire surface of the colon. So that is our idea. So we infuse it into the colon and shift them to the right side. So then it goes into the ascending colon and entire colon is painted. And it has to be done on a regular basis because it gets washed away. So if we do it for 2 to 3 months, this microbiome make its way to colonize there, and once it is colonized there and it does immune priming of the immune cells, then it keeps surviving and it can be sustained throughout life.
Okay, because these children are, when you're doing it in a colon way, these children are a little bit, it is a challenge for them. Do you do that in anesthesia or?
We don't give any anesthesia. We don't even restrain also. It's a painless procedure. It's a simple thin tube that we insert. So first 1 or 2 days only they resist. Once after they understand there is no pain or nothing, there is no major discomfort for them. And many children also feel better when we pass the tube, because when we pass the tube, all the gases come out and they start feeling better. Most of the children have these bloating issues and everything. So when we pass the tube, they feel better. So generally they don't resist after 2 days.
Okay, thanks, doctor. Thank you for the clarification.
Yeah,
I have
Hello, sir. Hello.
One second. We have a few questions on the chat, so let's just address them first. I request everyone to mute their mic and switch off their videos for these questions. So, sir, someone is asking, is it a one-time treatment or multiple years we need to come?
So it's so most of the times if you do it properly for 3 months, generally it won't require regularly. But some parents do come but when they come in the second year or third year, they come for a small period like 1 week or 2 weeks for the maintenance doses. So it can be done as a maintenance doses. You can if the child is comfortable and doing well. No need to come back and do it.
Okay, so the next question is what kind of screening you perform for donors and how do you ensure that the sample is always in good shape? Do you have enough donors all the time?
So finding donors is a bit difficult. So we currently have two donors, and we tested. So we test them for food intolerance test and their stool microbiome test and their routine stool test and routine blood workup. We do it, and they will be evaluated by our psychologist and evaluated by a pediatrician. And every day we will be in contact with their mothers, and when every day we see their specimen also, and we compare with the Bristol stool chart. And every specimen we do a microscopy test in our lab. Then only we use it, and we don't store. Though we have a storage facility of -20, -80, we use on the fresh samples on a daily basis. And we every day we follow up with the mother and we see the child and see the stool quality and do a stool microbiome. Every now and then, like every 6 months, we keep testing them for the routine blood test and other things. And if they have any sort of infections, also we keep seeing everything. And that's how we make sure that there is no contamination there is no transmission of infections.
Okay, so I hope that answers the question. The next question is, Doctor, which lab is best for getting food intolerance tests done in Hyderabad?
So there are multiple labs, like Dr. Lalpath Labs is there, Ampath Labs is there, Patkin Labs is there. Any of the good labs, NABL approved labs, you can do it.
Yeah. So the next question is, will you give anesthesia every day for procedure?
No, there is no anesthesia involved. There is no restrainment. So it's a simple.
Sir, hello.
Yeah. Hello.
సార్ ఇప్పుడు ఏలూరులో ఏదో వస్తున్నారు స్టార్ట్ చేస్తానంటే ఇది చెప్పారని సార్ మా ఫ్రెండ్ కి ఏమన్నా ఉంది అది ప్లాన్
సో ఆశ్రమ్ మెడికల్ కాలేజ్ ఆశ్రమ్ మెడికల్ కాలేజ్కి వచ్చి ఉంది అక్కడ కొన్ని కేసెస్ ఉన్నాయి బిగ్నిస్ మస్కులర్ డిస్ట్రోఫీ కేసు ఒకటి ఇంకొకటి అదే పార్కిన్సన్ కేసు అట్లా కొన్ని కేసెస్ ఉన్నాయి అవి వాటికి చేయడానికి వస్తున్నాము నెక్స్ట్ వీక్ అట్లా అప్పుడు కలవచ్చు.
ఓకే అండి.
Okay. So the next question is colonoscopy done every time or just once?
No, we don't do a colonoscopy. So I just mentioned because of the colonoscopy. Like colonoscopy has one advantage that you can visualize the bowel cleanse, and you can place the specimen at particular place. So, but it cannot be done on a daily basis. If FMT does not work with one session or 2 sessions or 10 sessions. It needs a long course. So colonoscopy option is not there for a daily procedure. So we cannot do that procedure on a daily basis. So we don't do a colonoscopy. We use simple tubes of 60 centimeters to 100 centimeters. We just insert through the anus. So we cannot see whether if it is going to 60 centimeters or 100 centimeters. But we can pass the entire tube. Most probably it enters into the transverse colon, and then we put them in the right lateral position. Then whatever the infusion we give, that enters into the ascending colon and paint the entire surface of the colon. This is our this will be done on a daily basis, and there is no coloscopy involved. There is no sedation involved, and the procedure is painless.
Okay. So the vitamins mentioned is best taken as infusion or tablets.
So infusions are the best one because when we are taking, most of the people will not absorb properly, and the glutathione injections or vitamin C injections or vitamin A and these things. So infusions are the best one. Like IV injections are the best, and some tablets will be there like astaxanthin. Those are not available in the IV forms. So those are the some things people can take it orally, but few infusions are necessary to remove the chemicals.
Okay. The next question is, hello doctor, can we have this treatment performed along with stem cell treatment? For example, one performed at Neurogen in Mumbai.
Yes, you can do it parallelly. So once you do FMT and stem cell or stem cell and FMT, you can do it, no problem.
Okay. The next question is, do you do parasitic test for the donor?
Which one?
Parasitic. Parasitic. Parasitic.
Yes, every day. That is done on every day basis. Like we do a stool microscopy to see any OR parasites are there, because these children will regularly go to the play areas and parks, and they play with mud and everything. So we do it on a daily basis. Every specimen will be tested for the parasites.
Okay. Can you share the food intolerance test name so that overseas patients can know if the children are even eligible for FMT?
Yes. So there are some tests in US. Recently, few doctors in US suggesting, they call it as food sensitivity test. Or so some test is called KBMO test. So I don't know much about their labs there. But if you ask for food sensitivity test or food intolerance test, you can call it as IgG based. Then people will understand. Those lab people will understand. You can do it there.
Okay. Someone has mentioned my son has failure to thrive. We done last month endoscopic and colonoscopy. They
Hmm.
identified both are good, but EOE mild to moderate.
Okay. Eosinophilic gas, esophagitis. Okay, got it. So eosinophilic means like it's an immune flare-up. So probably it can be recovered. Immune system can be changed with gut microbiome transplant. This kind of inflammations can be handled with FMT.
Okay. So actually the next question is, can we get the list of labs in India? I am not from Hyderabad.
Okay.
We'll share the list on
Yeah.
the email with everyone.
Okay.
So I'll move on to the next question. Can we do IV chelation of metals parallel to FMT? If yes, wouldn't chelate / excrete good microbiome?
No, microbiome will not have effect on the IV chelation. You can do IV chelation for the heavy metal detox and come for FMT. It won't be a problem.
Okay. The next question is, how long the process takes each day? Once the treatment is completed, any testing or evaluation to be done to make sure kids are cleared 98 to 99% before discharge from your clinic?
So it's a 1 hour to 1.5 hour procedure every day. Uh, so, uh, so you cannot see the food intolerance changes within 3 months. But so one, you do a food intolerance now and do a 3 months treatment, and then we have to repeat the test at the 6th month, ninth month, and 12th month. You can see the clear decline of the food intolerances there. So that the food intolerance test report will tell you the tale, the microbiome has changed or not, and the immune system has. Like immune modulation has happened or not. So and we can see the cognitive improvements and improvement in the sensory issues and fine motors and other things. Those are all with observation we can see. With assessment therapists and others, whoever observe, they also can see those changes.
Okay, so the next question is the charges fees are same for all kids or it depends on age, location, child severity? I think it's the same. There is no difference. Yeah, I can answer that. The next question is along with glutathione, what all can be given to remove toxins?
It is mainly N-acetylcysteine. It's a glutathione precursor. So glutathione is the ultimate scavenging molecule. N-acetylcysteine also act it's a glutathione precursor. Sometimes in some studies say that glutathione directly it will not enter into the cells and it will not scavenge. N-acetylcysteine it can enter into the cells and there it can form the glutathione. So it can with N-acetylcysteine giving N-acetylcysteine automatically will does the detox process.
Okay, so there was a short discussion in the chat amongst participants about the Arizona State University doing a similar procedure with or without capsules. So, what is the difference between them and us?
So Arizona State University, they're also doing fecal transplant also, and they are doing capsule form also. Capsule form, in the capsule form, what will happen is their stomach acids and pancreatic enzymes will kill it, and the fecal transplant must be same here and there. Probably ours is they might be taking it from the high diversity individuals, their focus is mainly on their donors are adults, our donors are children, and their main focus is on the metabolites. Our main focus is on the epigenetics. This is the difference between Arizona State University studies and our studies.
Doctor, I just want to add about Arizona State University, which I read their research. They also do, through the enema process, they do a bowel cleanse. The only difference in yours and theirs is they do an antibiotic treatment and then they do the microbiota transplant. And they also talk about an 8-week period to see the gains. And then the gains sort of sustain is what they've said
Yes.
in the research.
So we used to give vancomycin or rifaximin earlier, but what we observed, these microbes have much more stronger antibiotics than this vancomycin and rifaximin. The healthy microbiome has this. They have quorum sensing mechanisms, and they can identify what pathways the other bacteria has. They can target those bacteria and kill them. So the microbiome has better antibiotics than the man-made ones.
Okay, so the next question is does mold slash mycotoxin will reduce after this FMT, or do we need to also take antifungals in parallel?
So if there is a high antifungal infection, there is high fungal infection is there. So using antifungal treatment and with that doing FMT treatment would be optimal kind of thing because to transform the immune system, FMT will take longer period. So meanwhile, this fungus keeps on fighting the new microbiome, and it will impact on the microbiome survival as well. So in those cases, those have fungal infections and other flare-ups. So they should be given antifungals and then do the FMT.
Okay. The next question is, Doctor, the result of food intolerance test is valid for how many days, or the intolerance is forever?
So it is generally forever. If you are not intervening, if you do a FMT, then automatically it will come down. That you can see in the blood test.
The next question is, will FMT help with reduction of heavy metals and parasites since parasites feed on heavy metals?
It will help on parasites. FMT will help on parasites, but so FMT improves the detox pathways, but complete removal may not be done with entirely done with FMT. We have to actively do it with antioxidant therapies or chelation.
Okay, sir, Arizona University, the discussion we had before, is going through FDA process. What's the equivalent for approval of your research in India?
So they are into the phase two, and their phase one is already they have published it, and they are going to get it's a news that they are going to get a FDA approval for off-label treatment. So in India, we are going to publish, we are going to submit it to the ICMR, and ours is an academic study. ICMR also might approve in future once FDA approves that Arizona study.
Okay, so for the approval, so meantime, what are the procedures that are being followed at your end?
So we have found one ethics committee, and our institute is also getting registered as a research entity, and from all the procedures and other things will be reviewed by the ethics committees. So that's how we can do all these things.
Okay, any side effects during procedure or after treatment, such as hyperactivity or other sensitive issues? Any
Yeah.
side effects or during the procedure or after the procedure, after the treatment?
Yeah, in the beginning days, we can see die-off reactions. Die-off reactions means So I told spoke about biofilms and bad microbiome. They produce certain neuroactive metabolites, right? So when we are transforming, when we are giving the new microbiome and trying to kill the existing microbiome, those microbiomes, when they get destroyed and release those metabolites which enter into the stream, that can cause some aggressive behaviors in the beginning, or some other die-off reaction, like allergic reaction, the other things. Those things are possible in the first few days, and these things will settle over time. And other things like hyperacidity and rashes because we are doing on a daily basis, because the area is wet. Sometimes redness will be there, and some cracks can come, and some colic pain can be possible, and bloating and gas formation can be possible. These are some of the simple problems they can get, and those can be handled with simple medications.
Okay. The next question is, what is the benefit of doing it for 2 months versus doing it for a shorter period like 1 week?
Shorter period will not be helpful because the new microbiome will not survive. So shorter period may give you an impression that it is working, but it will not survive. So the new microbiome will not survive there, because our own immune system is primed by the existing microbiome. Our own immune system will fight back this new microbiome, and it will not survive there. So that's why it requires a longer period for the colonization of the new microbiome.
Yeah. So then the next question is HBOT ozone therapy helps after FMT?
They are mainly HBOT and ozone. They mainly act like antioxidant and giving more oxygen to the tissues. FMT, before FMT or after FMT, there is no role there is no change for HBOT and ozone.
Okay. The next question is, do you suggest stem cells during or after FMT treatment? If after, how much after?
So generally, there is one study. So people responded to stem cell therapy better after FMT. If somebody is using antibiotics or something like that after during stem cell therapy, don't do it immediately after that. Maybe take 2 to 3 weeks after FMT.
Okay. The next question is, my son also has high lactate levels along with, I'm sorry, I cannot along with aspergillus will have any benefits in reducing lactate levels?
Maybe it may be possible. We never tested for this before and after, but we can always see. So we do the FMT and then we see check the lactate levels and see whether they have improved or not.
Okay. Do you guys provide any other therapies during 2 months stay, like speech or occupational therapy, etc.?
Yes, we have a school kind of setup where specialized patient all therapies are there.
Yeah, that's about it. If we'll just wait for maybe another few minutes. If anyone has any questions, please, you can mention it in the chat column. Yeah, there's a question. Do we have any other centers other than Hyderabad? Not now.
Not now. Yeah.
Okay.
Because
Doctor.
it's six committee and other things. Right now we are only doing it in Hyderabad.
Okay. Doctor, do you suggest first FMT and then stem cell?
So it is based on one study. I can't figure it out which one to do first, but we can do it. So after FMT, I think. So I have thought process that the stem cells that you give, if you have your microbiome is not good and your epigenetics is not good, and you give stem cells, that stem cells can have the same epigenetic errors. Then they can go into the same epigenetic errors and may not work. So, if you correct your microbiome and correct your epigenetics and use stem cells, that may be better theoretically. This all theoretical and my own thought process. But what is in reality, we don't know.
Okay, so next question is, Doctor, FMT is only for autism kids or any syndrome kids?
So it is like FMT is like I told you earlier as well. Like if somebody is having deficiency in iron, you give iron injections. Somebody is deficient in vitamin D, you give vitamin D. Similarly, if somebody is having microbiome abnormality, any conditions, be it multiple sclerosis, be it Parkinson, Alzheimer's, Crohn's, or ulcerative colitis, Clostridium difficile, inflammatory bowel disease, irritable bowel syndrome, those who have food intolerance positive, it can be done.
Okay.
Can you please email the
Yes.
name of the test for foreign children or foreign patients, please?
Yes. I replied on the chat that yes, we will do
Okay.
that.
Thank
We
you so
will do
much.
that. We'll send it to everyone. Yeah, and just to add on to a question before someone mentioned about if there are any other therapies available. So basically, at Risprise Institute, we have a whole child development center which has available. It's an entire setup where ADL therapy, speech therapy, behavior therapy, occupational therapy, sensory integration, ABA therapy, psychological intervention, special education, group therapy, play therapy, art therapy, everything is provided. And all of this is under one roof. So it also has different patterns. You can, the child can come from for 6 to 8 hours. The child can be a part of it for 4 hours. The child can be a part of it for 6 hours. So it is a huge setup wherein the child will be taken care of and all the therapies will be taken care of. Education wise or therapy wise, nothing will be missed while your 2 months stay at Hyderabad.
That's great.
I'll just go through once more if there are any additional questions.
Is it you guys doesn't have any accommodation or something we have to find ourselves? Is that right?
Correct. So we can help
Okay.
you with the service apartments at the hospitals nearby. We can connect you with a few agents. But we don't have anything in-house because it's not a IPD procedure wherein you have to take admission.
Is it walking distance from your clinic?
There are many service apartments and hotels who are at walking distance as well.
Okay, great.
Yeah. So you can choose. You have options.
What is the next process like? What is the next step? How do we reach out to you guys to, book everything?
Yeah.
If
So myself is Siksha, and I have my colleague with me as well in this Zoom. His name is Karthik. One of us will get in touch with you through email and through WhatsApp.
Okay, great. Thanks.
Yeah, we'll send all the information. We'll send the reading materials. We'll share the YouTube link of this webinar, and we'll share the test details also. Someone was asking for it. So all the requirements which were mentioned in the chat column, everything will be shared with you on the email, on the first communication from our side, and then we can definitely take it forward.
Great. Appreciate it.
Yeah. Sir, do you want to add anything?
So nothing from my side. Any questions? Any other questions?
I have a last question. I've heard like if you like do enemas or if you do anything with the anus or rectum, the elasticity gets very loose. Is that true, or will that be affected with this procedure at all?
No, we didn't see such kind of things in last 4 years.
Okay.
We never saw that such kind of things. Yeah.
Okay. Thank you so much.
So your video is turned off.
Okay.
Any yeah, we have one more question. Doctor, does polymicro polymicrogyria come under brain structural defect? I don't know if it is gyria or gyria.
Yes.
Yeah, I'm sorry for my pronunciation.
Yeah. So it's not microarchitecture. It's a gross abnormality.
Sorry, come again.
It's a gross abnormality only. It can be picked up in the MRIs.
It comes under the brain structural defect.
Yes. Yes.
Okay. And is the doctor will the doctor take procedure for all the children, or any assistants will do?
So it can be done with assistance only, and it can be the parents also can get trained. It's a simple procedure. We even do parent training. If somebody's sibling or some child has a sibling or some cousin who pass the donor test and everything, parents can get trained and do it. And it's a simple procedure. Nurses can do it, and the parents also can do it. Only very few cases require doctors and specialist assistants.
Okay. And is the procedure safe?
Yes, it looks safe since last 4 years. So provided the process is correct and the donor and everyday so if the child is monitored, donor child is monitored, it will be safe.
Okay, to the question of polymicrogyria of the brain structural, the additional question is, does this treatment work for that condition?
It will not repair the structure. So it's not even repairing the microarchitecture, right? So this is a gross abnormality only, and it cannot repair. FMT cannot repair the structure of the brain, but it only can bring the chemical balance, brain chemical balance. The neurotransmitter balance can come with the FMT.
Okay. My son is aggressive. Can we opt for this procedure? Will he cooperate?
So first few days, he may not cooperate. It might be difficult. After a few days, like if most of the children with aggressive behaviors, if you do a proper bowel cleanse, so their microbiome is the reason for their aggressive behaviors. If you reduce the load of their microbiome and wash out their entire stool, automatically their behaviors come down. And some children, even after bowel cleanse, they still exhibit aggressive behaviors. For them, we use some kind of enemas like alcohol enemas and nanocurcumin enemas. These kind of things we do to suppress their microbiome and give the new microbiome. And that's how we try to control their aggressive behaviors. So when we give nanocurcumin enemas or bowel cleanse and followed by nanocurcumin or alcohol enemas, then we usually see there is a change in the aggression in the children.
Yeah, that's there. Okay. I think, sir, if you can just give like a short conclusion and we can end the session here. If anyone has any further questions, I believe you all must have received my reminder messages already. And our email ID is also with you guys, so you can get in touch with us for any further queries. Over to you, sir, for conclusion.
The gist of the entire session is like the environmental chemicals are the primary cause for autism, and the microbiome disruption due to secondary to environmental chemicals is aggravating the problem. So replacement of this microbiome is necessary when there is a disruption in the microbiome for these children, and parents who have autistic children, they can go for when they're going for second child, they must detox themselves before pregnancy and maintain the detox status throughout pregnancy, and they have to do a lifestyle modification and reduce their chemical exposure to avoid autism in their child. So this is our entire work in the field of autism. Thank you very much.
Thank you everyone for the participating, and we'll connect with you for if you need any kind of personal one-on-one consultation and discuss about the case specifically. Okay, bye-bye.
Thank you.
Thank you very much.
Thank you.